Thank you all very much for coming. I'm glad to see the room being this full. It's very nice to have you all here. So today we're going to be talking about equality in AI-driven drug discovery, and specifically we're going to focus on rare drugs and rare targets, and if AI can help to design drugs for rare disease targets. So we'll have two speakers today, and Paula and Hagen, they're sitting over there. And I'm going to introduce them. Paula will talk about rare diseases, and then Hagen will give us a talk about AI-driven drug discovery. And then at the end, we'll have a discussion, an open discussion, I hope. So feel free to engage and raise your hands and ask questions. And we'll kind of like spitball ideas around how AI can be used to design drugs for rare diseases. So we'll leave a little bit of time after each talk for a short Q&A, but then bear in mind there will be this bigger discussion at the end, so you'll have more time to ask questions. All right, with that, our first speaker will be Paula Fernandez Guerra. She's sitting over there. She is a senior researcher at the University of Southern Denmark. So she came all the way here from Denmark. Thank you. She works in personalized medicine, and she is working on developing adequate therapies for patients with rare metabolic and mitochondrial diseases. She was also a chairwoman of the Society of Spanish Researchers in Denmark and a vice chairwoman of a network of Spanish researchers abroad. She's originally from the sunny Canary Islands, so hopefully she's enjoying the sunny day we have in Oxford today. All right, Paula, welcome. Thank you.
Thank you so much for the introduction and the invitation. I'm very excited to be here. I'm enjoying very much the lovely weather. Hopefully in Denmark it's raining as it should, so they can be very jealous. So, I'm also very excited to talk about this topic. I'm very passionate about rare diseases. I have been studying rare diseases for 15 years now. So, but what are rare diseases? No, of course. Something. Yes, it always happens. So rare diseases are actually duckling of biomedical research. Quacks like a duck, swims like a duck, is not a duck. And that makes a huge amount of problems because we cannot just pipeline the patients as you usually do with more common disorders. And that means that they are usually misdiagnosed, mistreated, and not supported. So how can we help them? Then let's ask them, because no matter how many years you spend studying a disease, nobody knows more than a patient that lives with the disease every single day what they need. So this is a consortium of rare diseases in Europe, and they have made a survey with 4,000 participants that includes patient caregivers and patient representatives, and what do they want in the next year to happen. So this is what they want. Leaving no one behind. And that's the key. Independently of how frequent your disease is or how difficult or strange or complicated, that doesn't mean we should give up. Of course, we have an issue of cost effectiveness, and then I'm wishing that AI will solve all those problems, or at least it will help us to make this a more efficient process. Just some numbers, there's 30 million people living with a rare disease in Europe and 300 million worldwide. So maybe they are rare in their own disease, but collectively, it's a lot of people. Worst part, there's no cure for most of them, and there's very few treatments available. So they're very debilitating disorders, and it's very sad diagnosis to give. We look at the UK, there's 17 people have a rare disease. This makes a huge burden for the public health system, because it's also very debilitating disorders, meaning they will go a lot to the hospital, they will go a lot to the doctor. They also usually don't work because they are disabled for that, meaning they don't produce. So if you don't want to treat rare diseases because it's the right, as every person has, do it because it will save you money in the healthcare system and it will put money into the economic system. So pick your evil, I don't care, let's get it done. The diagnosis of a rare disease in the UK, it takes from 4 to 6 years, and that's kind of the average in Europe. So it's a long time for a patient to just go from doctor to doctor until they find what's going on. not only they feel isolated and unsupported, they are also being misdiagnosed, and sometimes they're given a treatment that actually makes them worse. 80% of the rare diseases have a genetic cause, so we need to invest in not only finding which is the variant happening, but what that variant is doing. And of course, ideally, genetic therapy will be there, but what can we do for them today? 70% of the rare diseases affect children, and 30% of those will not reach their 50th birthday. So it's not a debilitating disorder, it's also a life-threatening disorder. But what do they want? 80% of them, they don't expect a cure in the next 10 years. They have given up on that. What they're hoping for? They hope to have the disease stabilize, and they hope to manage their symptoms if they're progressing to progress slowly. All in all, a better quality of life. And that's what we can help them to achieve today. And we can do that using personalized medicine. This thing of one drug for everyone, it doesn't work, especially in rare diseases, when even if you have a mutation in the same gene, they can be very different from each other. So we need better diagnostic tools, better biomarkers, and a combination of drugs and therapies that can help them. And that's where my wish list comes, and I hope AI can solve all those questions, or at least help us prioritize. Because when I get to a patient, we say, okay, we have found out where you're sick. We say, okay, what can I do? I don't know. And there's a lot of drugs out there, there's a lot of therapies, not only medication, it's also physical therapy and a lot of options there. How can we do that? So my wish list, and maybe Hagen will see if that's possible, is can we rank them? Where do we start? Instead of just, going through the literature. It's rare diseases, so there could be a case from the 80s that you have to follow up. It takes a lot of time. I'm specialized in metabolic disorders, and mitochondria is what covers 80% of our metabolism. Go back to high school, you have heard about it, you have forgotten that it's there, and it's what produces most of our energy. So the most cause is fatigue. That's the main symptom that is common to rare diseases, both mentally and physically. When we talk about fatigue, a lot of people think, I'm so tired on Monday mornings, this is horrible. But a patient with a rare disease that affects the mitochondria cannot hold the arm like this for 30 seconds. So we're talking about extreme fatigue. They cannot read because they cannot focus so much to read. So it's very debilitating. And instead of just talking about biology, I will show you 2 cases of what we do in the labs and where I hope AI can help us. So a few years ago, we were approached by physicians from Canada saying that they have a patient that they suspected has a metabolic disease, but they didn't know. So the story of this young boy is that at 20 months old, he was walking independently. So it was a big delay in the development. By 3 years, he was clumsiness, gained stability, frequent falls. So at this point, the pediatricians were thinking, this is not just a slow development, this could be something else. So they started some clinical examinations. For years, they did a brain MRI, and they observed hypomyelination. Myelin is this sheet that covers our axons, and it's very important to protect them because the signals come from our brain all over our body. So myelin helps that this goes to where it should be. If you think about a cable, it's the folder outside that avoids that you have set on fire with a shortcut. They also find out that they have elevated malonic acid, which is a mitochondrial metabolite, and they did NGS and found a variant in the HSPD1 gene, which encodes for mitochondrial protein. So all of that, they make us confident, okay, we suspect this is the cause. But they were not so happy because by NGS, they found only 1 copy that was affected. So they were, is this doing anything? Nothing? We don't know. So okay, fine, let's just give us some cells and see what can we do. So HSPD1 encodes for this protein, HSP60, which is a molecular chaperone, which are very fascinating molecules that they were molecular proteins, that they assist the folding of other proteins and make them active. In this case, HSP60 folds 300 out of 1,500 proteins in the mitochondria. So if it's not working, it's a lot of things that are not happening. When we look at the mutation, we could see that not only this mutation, but all the mutations in this protein are in the same area. So they have these 2 subunits, and it's the join in between. So we start thinking, if there's a change there, it's probably not going to work. It's also highly conserved protein, so very few amino acids are compatible with life. And if we look from the top, we could see that they're all in that ring inside, and that's where it connects with the protein that they have to fold. So if there are some changes there, maybe it cannot do the folding. And it's also very close to the ATP that you can see there in red, which means maybe the hydrolysis of ATP that needs to happen, it won't happen. So all of these make us think, okay, this patient with the mutation leucine 47, this one in green over there, will it work or not? But does it depress the protein? It doesn't express, nothing happens. So that's the first thing we did. We quantified how much of the mutant protein is there. We did it by mass spectrometry, and as you see, the control had 100% of the wild type in blood, and the patient has 50-50. So the mutation is expressed, and the mutation remains there. So now what about the activity? So we do an E. coli complementation assay, where we have removed the homologue of this protein in E. coli, and have a plasmid with the protein wild type, as you see in black. And here you see what happens when we have the mutation. There's no survival of the colonies, which means it would be very hard that this mutation works. So that's what we know. This is our neuron, and this is one of the things that could be happening. The mutation is not working, so maybe there's a developmental issue and the axons are not properly formed. But it could also be that the axon is properly formed and the myelin is being degraded. That would be similar to ALS. Or it could be that both the axons and the myelin is being degraded. And you may think, okay, what matters? You know what's the cause? But the treatment, the prognosis for each of these models is very different. So we need to know a little bit where is it going. So then we have used now patient cells to know what's going on. We developed a cellular model using CRISPR-Cas, and the same with an animal model. But all of this takes 5 years, and a full-time PhD student and master student. It's a lot of work and it's a lot of money, and we still cannot tell the patients, yes, you're sick because of this. So, okay, what can I do? I don't know. Because also this is the answer of our research. You know what it is? It's just every single big arrow is something that is upregulated or downregulated in these patient cells. So when I look at this, it's like, where do I start? It's so many things. So this is some of the parts that maybe with AI we will figure it out. Okay, you don't need to change all of them. If you change these 2 things, you will change a lot of them with it. And maybe we can skip this part of having to spend 5 years trying to figure out what's going on. And we can have already some predictions of what could be. And I mean, a zebrafish is still a zebrafish. What happens in the person is a bit tricky to do. So that's my wish list for the first case. The second case is mitochondrial DNA mutation, because in case you have forgotten, mitochondria are so cool. They have their own DNA, translation, replication machinery, everything. We cannot live without them. We come from our moms. So that's to say something. This is the most common mitochondrial DNA mutation, which affects the tRNA for leucine, which means all the proteins encoded in mitochondrial DNA are going to have a defective translation. It's a very heterogeneous disease, runs from asymptomatic, people who not show symptoms in their life, people who have mitochondrial inherited diabetes and deafness, MIDD, which is the most common phenotype, and others who have what's called MELAS, and that's how it's more commonly known, which is mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes. This is a very debilitating one. This usually ends up being housebound or bedbound. In my case, I'm not working with those symptoms. We were more focused on the diabetic part and the symptomatic one, because those people are now starting to get into their 50s, into their 60s. These diseases are all very new. The first cases are from the 90s. So now they are surviving, fine. What happens when you get 50, 60, 70 years old? You have bone issues generally, just in the normal population. There's osteoporosis, there's a high risk of fracture. So what about these patients? Do we have to worry more about them? Do we have to scan them regularly? Do we have to do a preventive treatment? We don't know. What we know is that a bone fracture for these patients is very severe because they will make them not move. So the bodies will degrade a lot faster than the normal people. And they will probably not recover the muscle they have lost on the way. But also some of them have such severe cardiomyopathies that they will not survive the surgery to put a metal rod, which is the case with one of our patients. So what's going on? We decided to study that, and we took bone marrow aspirate and we studied stem cells from the bone marrow. Because I know you all have forgot about high school and undergrads. This is basically what happens with energy metabolism. We have fatty acids, amino acid, and glucose that they will go into the mitochondria and produce a lot of ATP. Then we can also get ATP through lactate production from glucose, which is very little, but it's very fast, so it can compensate. So what happens if this is not working? So we went to study that in those cells, and we could see this diagram. In the y-axis, you have mitochondrial respiration, that, as you can see, is decreased. The yellow is the patient, the blue is the controls. And in the x-axis, you have glycolysis, that, as you can see, is increasing the patient. So they are compensating, but are they compensating enough or just wasting resources? So we measure where the ATP comes from, and we can see that they're compensating enough. They are not losing ATP, but they have a metabolic shift, and they're producing mostly ATP from glycolysis, which initially doesn't mean much, but that means that all the byproducts of metabolism from the mitochondria are not happening, and that could affect the bones. But what can we do? We don't grow bones in the lab, sadly, that would be very cool. So we use a mouse. We have immunodepressed mouse where we have bone marrow stem cells from these patients, and we have hydroxyapatite as a scaffold, and we have a subcutaneous implantation. We have them there for 8 weeks, and these cells will form bone, or at least initial stages. We retrieve the implants and we do an H&E staining, as you see. Yeah, it's a funny purple, but kind of purple. Is the bone, the gray is the hydroxyapatite, that's the mineral, and connective tissue is the pink. So if you look at the results from this patient, there's a very big difference between the control and the patient. So this patient has, 50% capacity to form bone. Then that's in the mouse and the cells, okay, that will happen in the whole organism. The only way we have to see is to take a bone biopsy, and we will see that there's a 50% decrease bone density. So it seems that something is happening, and it seems that something with this metabolic shift. But here's when we start getting lost. Luckily, there's a pharmacological company, Condrium, in the Netherlands, which are developing treatments for this specific disease. They're not looking at the bones, but we thought, okay, we can collaborate with them and see if this is a treatment we can do. So they're working with this compound, I will never be able to say it, sonicromanol, which is already in phase 3 for treatments with this mutation, which is something nice because we can tell the patient you can use this now. You don't have to wait 10 years until they start to look. And it's a reductive and activity stress modulator and anti-inflammatory, which is the main consequences of this mutation. So we will use the active metabolite AH183 with our cells. And as you remember, they have a metabolic shift in the energy metabolism that we could rescue with this treatment. As you see in purple with the treatment, and the vehicle is in yellow, there's an increase of mitochondrial respiration and a decrease in glycolysis. So the patients before were here in the lower right quadrant, and now they're in the upper left, where the controls were. That means our treatment, at least in vitro, is working. We also look at the ATP, and we will see that we have reversed the shift. And now the patients with the treatment is approximately 50% ATP from the mitochondria. Will that improve the bones? We don't know. How can we know? We have used the patient cells, we can have a cellular model, and we can have an animal model. Getting to the point where we are now has taken us 3 years. Getting to here will take us another 2 or 3. So can AI make my life easier? That's the hope. Especially to have a bit of prediction and to say, because this is one of the drugs I could have tried another hundreds, and maybe it doesn't work alone, maybe it works better in combination with others. That's too many things to try. So maybe with AI, we can run them and make this a more cost-effective process. And then I can go to the economy people and tell them what to do. But just to finalize, I would like to thank mostly the patients and the families and all the participants in the clinical studies for giving their time and participation, and my colleagues from Ocean University, where I did the neurological study, and my current workplace can be in Odense, where I'm doing the bone things. And if you have something to remember, just let's leave no one behind. Thank you.
Thank you very question?
Question over there. Yes, when you say you want to make your life easier using AI, what realistically do you think is a stage that AI can get you to, if that makes sense? Like, it definitely can't do everything, and there's still some testing that needs to be done in the lab. But how much can it?
So that I don't know. My hope is that instead of me going through the literature and thinking what's this treatment best or this one, can give me a rank and say, of all the treatments available today, you should start with these 5. That's more doable because this is a huge risk. Of course, there's some, they have tested in muscle cells, in others, so there are a lot of good indications, but indications are just that. So I will ask some more robust data. And especially I would like that it show me things that I cannot see. When we have all this complex from the first patient, maybe there's a drug that it can help me to fix a lot of them at the same time. I don't know that, or it will take me a lot of time to find that. So that's why it could be more cost effective.
Any other questions?
Collaborated with any, sorry, thank you for the talk. Have you collaborated with any constitutional AI people in your university? Is that feasible?
Not currently.
No.
It could be possible, and this is something that is coming right now. So we still do it in the old school way. So we're testing everything in the lab, but we're definitely considering it because these are the success stories. We still have easily 50, 60 patients undiagnosed. So maybe we can streamline that process. That's would be a nice.
Question there at the back.
Hi, I'm not a biologist, but could you explain why you chose to use the mouse as your model organism? And what are the advantages of mice over other potential?
Well, I would love not to use any animals to start with. That will be my dream. The thing with animals is that right now with this mouse model has been tested for the last 10 years to see how the production of bone is. And that's basically why we know it works. And if something goes wrong, it's not because the model doesn't work, it's because my patient cells doesn't work. So I would always, and I don't have that many patients. This is a bone marrow sample. I don't want to take more of those. So my cells are very precious for me. So I would always use the most robust method I can find. And in this case, it's the mouse model. The mouse model is immunodeficient, so it will not react to the implant. And we also know that there will not be cells of the mouse in the implant. So everything we have is human bone.
I was going to kind of ask a question maybe on a different level. How do you choose which rare diseases to kind of study? Is it based on what's most promising or like just sort of you're feeling on behind?
Yeah, it's whoever gives me money. That would be the sad truth. What we also try is to group them together. So with this patient with a neurological disorder, it's not the only one, we have a few. So we try to get knowledge from one that we can apply to more. Also, you think about the rare diseases are very extreme, so we can also gain knowledge that we can apply to more common disorders, like these diabetic patients. It's a very different diabetes, but what they have shown us, we can apply to a large cohort, and that is easier for us to work. But sadly, it's whoever gives us money. And in that sense, the patient associations are doing a great job getting some funding that sometimes it's just the initial testing, and then you can see, okay, this is something we can do. Then it's easier to apply for large foundation. That particularly works very well here in the UK. They have been doing a great job at lobbying and getting funds for it.
Yeah. Do you know if anyone is working on a digital twin for mitochondria, or is that always complicated?
Studying what?
Like a digital twin, like especially like computational model formed.
Okay. I don't know. That would be nice, because that's also a bit of the issue. The prediction is very much limited on our own brain, so it would be nice to have a more system. Maybe it's there. I don't know.
On that note, can you have a sense of what proportion of rare diseases are related to mitochondria? Sounds like there's sort of this serious effect.
Yeah, so the proportion, that would be hard to say, but it's the ones I work with. So only there's quite a lot. No, but it's there. Some of them are highly frequent because they can balance each other. So with this mitochondrial DNA, you have more than one mitochondria in your cells. So there's a lot of threshold happening. So there's a lot of people who is very mildly affected or very symptomatic. So that's a bit, but in the global proportion, that I honestly don't know. But it's all the metabolism, so I would expect this at least half of them will affect the mitochondria primarily. Then you have secondary mitochondrial dysfunction that happens too.
So you talked about AI with like the more diagnosis aspect, but how have you like looked at AI? Maybe you haven't personally, but do you know of like AI systems to help like propagate the research? Obviously, our researchers like you are finding out, you know, these symptoms are caused by this. But then, like, is there propagation going back to help you select like groups of people to classify the rare diseases kind of together? Because it might be like groups in different ways that you didn't think that they would be.
I mean, we played a bit with this ChatGPT and giving some symptoms, and some of them it works, is something we knew, but others there was a bit of funny things. So it could be that they're completely crazy, or it could be that it's also telling us that this patient that we think belongs in this box actually belongs in other boxes. And at the end, they don't belong to any box. That's our brain limitation to understand diseases. But that could be a way, because if we can see that they fit in this box, then we could see what treatments are here. And maybe they need to combine. And that's for me what the potentiality is, because thinking about one treatment, that kind of I can do. But what about if they need 20% of this and 80% of this? How do we combine that without launching a clinical study, which is very difficult in this case?
Any other questions? Maybe we have time for
Yeah, thank you. I guess my question is about really again data. So if you want to build, I guess, an AI model to have you predict these things, from my limited understanding, I can imagine there's very little data, right, for rare diseases. And also I can also imagine that they're very distinct, like phenomena and how they happen. Are there like starting points from where we can get some sort of data sets that have of rare diseases as a generic concept?
I mean, if you think about most of the animal or cellular models, are extreme knockouts or removals of proteins, and that looks a lot more like a rare disease than a common disorder. So I will look to those databases where they have to take a mouse model that have studied up and down and far and left, and start there. Then you know what happens if this protein is missing completely, and that will be similar to a huge loss of function situation. But of course, some of them are very unique, and that's a bit tricky. But at least for the big streams, it could work in that way. Then it's the limitation of the gene editing. That's another issue.
All right. Thank you, Paula. So our second speaker will be Hagen Trendel. Hagen is the director of AI at Exatia. We also used to be colleagues, so I know Hagen quite well, and thank you very much for coming and joining us here today. Hagen has previously also worked at GSK. So I should probably say what he does at Exatia. Apart from being a director of AI research, his team works on developing ML models and machine learning models that help to find small molecules to treat different diseases or that would bind to different protein targets. And Hagen has also previously worked at GSK, where he led the development of the ML platform, and he also worked at a biotech startup in London called GTN, and he was a head of research there. So all that might lead you to think that Hagen has been in the field of drug discovery and ML since the very beginning, but don't be misled. Hagen is a physicist at heart, and he decided to rebrand, and now he's working in ML. So with that, Hagen, thank you very much for agreeing to be our speaker. Off you go.
Thank you so much. It's actually really a pleasure to be here. Thank you a lot for the invitation, and I'm looking forward to a lively discussion later. So I want to tell you about AI-driven drug discovery, and let me first tell you the problem. So drug discovery is incredibly complicated, and you can see this by the numbers of like what is the usual drug discovery project. So usually it takes like 12 to 14 years to find a drug. It costs on average $2 billion to successfully find a drug, and a lot of, for instance, a lot of it is that a lot of drug discovery projects fail. So I think 9 out of 10 projects fail in clinical trials. So that's essentially the complexity you have to deal with. And that's where you want to do better using AI models. So this is roughly what Accenture tries to achieve this. So there's a few principles. So we believe drug discovery is a learning process. So it's not a search problem that we try to find someone in chemical space the right drug. We need to learn iteratively to understand the problem better, understand patients better, and feed that back into the process until we get to the point where we understand it. And then the second part is to encode and automate drug discovery, to make it faster, to make it cheaper, and to develop better drugs faster. And then the last part is ultimately what you want to do is you want to do something, you want to cure a patient. So in the end, whatever you do needs to address that problem. So you need to link everything you do somehow to how these drugs will work for patient. So that's a large level overview of what Accenture does as a company. And a lot of it has some overlap with what Paula was mentioning about biomarker discovery, target discovery, etc., omics data. So generally, we try to essentially use all the data we get, incorporate it in our system, learn from it, and then drive it forward. So I will focus on the drug design part, which is essentially the piece in the middle here, where we essentially design drugs to make inroads. So maybe the other parts we can have more of a discussion later. Right. So essentially, how do we do drug discovery or drug design? We have certain data available. We define an objective, what we want to achieve with a certain compound, essentially a checklist of what good properties the drug should have. And then essentially we do iteratively the same process again and again and again. So because it's a design cycle, so we build based on the data predictive models. We use these predictive models together with a generative model to create new compounds. Then we rank these compounds and select the ones that ideally are as good as possible and give us as much learning as possible. So we use active learning. And then we synthesize, test, gather data, and do the same thing again. And essentially, the ideal case is we use this as efficiently as possible and get as much learning as possible so that we don't need too many design cycles. Right, and so that's roughly how we would structure a project. We would say, okay, this is a checklist. These are, so our designers would say, or our biologists, these are the properties that a successful compound should have. So it needs to bind and inhibit, for instance, the target we have selected has to have certain properties, how it behaves in the body. And then we code this mathematically into a function where we combine all of this together. And we can use this to check how a project is going. Are we still exploring and trying to find better compounds? Are we at some point in the case where we find quite a few good compounds and try to really drive home what we want to do? And then we can also use this in our AI models. That's very fast. Right. So let me go through this, what I showed you before, this flywheel. Let me talk about predictive models, what we do there, about generative models, and then how we close this with active learning, etc. Okay. So sometimes people say deep learning and modern machine learning methods only work with a lot of data. So that's not true. You just have to make sure you leverage it, all the data you have as effectively as possible. So one thing we find usually is that any benchmarks, any evaluation strategies in public space are not very helpful. So what we have to do essentially, we look at our own design data and say what models work best to predict on future design cycles the properties of compounds. And that's very different from usually in the literature evaluation or benchmarks are. And that also leads to very different models dominating what works best. So generally, we need to generalize much more out of the data set to new compounds that look different. And there we put a lot of effort into finding new methods to achieve that. So one is we build essentially good representations of molecules. So we can use data from computations, from quantum mechanics, from molecular dynamics. We essentially train models that represent the molecule in a very good way. And then we use, so there we have a lot of data. Then we use this data on our predictive tasks, and that usually achieves much better performance. And usually, so the other thing, how we try to get more data than what's usually accessible is we combine tasks, different tasks, so different properties, related properties, and we transfer models that work very well on large amounts of data to scenarios where we have much less data. All right, so let's say we have built some models. I told you that we actually care about what happens in a human, what happens in a patient. So we want to link very basic properties where we can measure in experiments to properties that are much closer to the human. So what we usually do is we predict these properties. We have uncertainty measurements for these properties, and then we combine them based on science and on mechanistic models into processes in the human body. And from this, we want to estimate what is, given these properties, what would be the dose for compound that we want to give to a human for the drug to be effective. And then we can compare this to toxicity effects that we predict, etc., to see how good of a drug is it, where are the problems. And because we also look at how sensitive it is to the properties we measured, we can decide which properties we have to improve more, and how severe toxicity effects are, etc. So that way we link like basic assays to like very complicated things like pharmacokinetics and human dose. All right, and here another example of how we try to build predictive models that work well, even if there's not much data. So let's talk about binding. So problem of, does a small molecule actually bind to a protein? And what we do there is we think of adjacent problems that are similar and where we have much more data. So for example, we can do a lot of very complicated quantum mechanics computations to understand how a molecule is moving in vacuum, interacting with another molecule, etc. And you can have a lot of data there. You can train a model that is quite accurate, nearly essentially as accurate as these computations. It's maybe slower than other methods, but much more accurate, and definitely much faster than these quantum mechanical computations. So, and then you can use this for molecular dynamics, but you don't have to stop there. So you give it to your colleagues that do molecular dynamics, and then you use this model just for your next task where you have much less data. So let's say docking your compound into a protein, right? And there you try to get a model that works there better than computation methods. And then again, you can think about a more complicated problem, like actually predicting whether this compound binds to the protein or not. And again, train it on very little data, and suddenly you have much more success. At the same time, for instance, if you use this in a 3D modeling, you can apply this to a lot of different targets. So let's say even if you have very little data on one protein target, you can learn from the other protein targets where we have done much more work. And hopefully this generalizes to this other case. All right. So that's what I want to say about building predictive models. And I think I'm actually a bit too fast. So now, generative models. I don't know who knows what a generative model is, but essentially a generative model, imagine an algorithm, you give a big set of molecules and you build a model that gives you molecules that look similar to this compound or to the set of molecules. And you just train it. And then you want to give it objectives like increase binding, make it less oily, etc., etc. And then you want this generative model to produce more and more molecules of the type you want. So it learns from all the molecules it produced before, gets feedback from these predictive models, And it tells you, okay, this is a good molecule, this is not a good molecule, and it will learn to produce more molecules that are of high quality to you. Right? So that's what we use in this case. And here's a bit more detail. So we have different models. Here's an example of one that's also very well widespread in the community generally. So here you first build a model that just generates molecules. So that's a classic generative model in AI. You train it on a data set of a lot of molecules that you believe are somehow of the right kind, that are good for this space. And now you build on top of that a reinforcement learning agent. So you have an agent that generates these molecules, but gets feedback based on these properties that I told you about before. So based on this list, on this multi-parameter optimization protocol that we defined at the beginning of the drug discovery project. Right? And then this agent continuously learns and gets better at generating these molecules. Another algorithm we have is called Gambit. So that's an evolutionary algorithm. So that's a very, let's say, much more classic. And that is based on you have a certain population. You evolve them, you give feedback, you combine them in different ways. And from this feedback, you learn how to get better compounds in it. That's a nice video. But what I want to show you is essentially you have scores on all these different properties with predictive models. You combine them, you say, okay, in total, this is a good molecule, a medium molecule, or a bad molecule. And then you learn from this iteratively. Right? And ideally, at some point, you get to the molecule that you actually. All right. So now we have predictive models. We build a generative model on top. We train it to produce molecules that tick as many boxes as possible. So what do we do now? So we have a lot of molecules. So now we have to select which ones we actually want. I told you in the end, we just take something like 20 molecules, a few dozen, and send them to the lab, test them, and learn from that. So we just try to pick those molecules in the ideal way so that we maximize learning and at some point find the best compounds that we can find. So how we do this is essentially we don't just predict these properties, but essentially distributions of these properties for each property. Then we combine them through these mathematical functions that I told you about before. So we get a distribution about the desirability or how good this compound is. And then we can decide, do we have a chance that this compound is really good? How big is the chance? And we can define again an acquisition function that just gives us a score. And by this we can rank these compounds. And then we can select a few dozen. And ideally, if we do this right, we pick them in a way that is spread out over the space so that we explore as much as possible. All right, and so you can do some experiments with this. You can do a bit of tricks. And then, for instance, here's something from the literature where people were testing on humans that were selecting compounds, and then on algorithms. And we essentially just added our algorithms. And you can see that essentially active learning algorithms completely outperform most experts. I think there's one expert that's very good, but in general, you're much better. All right. So now we have selected compounds, but there's a big step that also costs a lot of money and is very complicated. And that's a question, can we actually make the compound? All right. So you might have selected compounds that you will never be able to make, or that are extremely expensive and will take a long time. So ideally, you want to know about this beforehand, and you want to make it part of the selection process, part of this reward for the generative model. So you want to build tools that can predict how complicated a compound is or not, and that's very non-trivial. And ideally, we want to actually say how to make this compound, right? And there's different models you can build. So a very simple one is you say, what is the outcome of a reaction? So I take a few compounds together, I get an outcome. These models you can build very well. They have usually a very high accuracy. Now you can say, I have this compound, how do I make it? And it gives you a tree how to make this, right? But then usually those work much less. But now you can use this first model to check if this tree is actually correct or not, and feed that back into the algorithm. So again, from a simple algorithm, you learn a more complicated algorithm. And And then eventually you can essentially build a generative model, as I discussed before, but based on these trees of synthesis. So you can essentially say, okay, these are my starting points, now build me molecules, and let's see what their properties are. And then again, you can use reinforcement learning and can learn to make very good compounds in a certain number of steps. All right, and then there's things like actually predicting how to do these reactions. So that is also important for the following reason, because actually what you want to do is really close the loop. So you want to build an automated lab where you don't just tell humans, okay, make this, please, and in a few weeks I get it back, and then I test it, and in 40, 50, 60 days, I have some results. You want to make this as fast as possible. And this entire make test part of the cycle takes very long. So what we are essentially doing at the moment is building an automated lab that does this all very fast. And if you can limit yourself to compounds that are easy to make, you can spin this wheel very fast. And hopefully you get also more information and make drug discovery more effective. All right, so that's essentially my last slide. And here I want to just show a bit how this closed loop looks in a particular case. So that's a hit ID, which means essentially you're at the very beginning of a drug project. You have a target, maybe you have already some data, but you wonder about new starting points from which you can start. Yeah. So what we usually do is we just take small molecules, very small molecules, so we call them fragments, and have an assay where we, let's say, wash them over a protein that's fixated. We can measure what binds, and we get data at a large scale. So very noisy data, but we get a good starting point. And then we, again, use a generative model. We elaborate on these fragments with bigger molecules. We score them with based on the data we have already. We select with active learning, again, the compounds that are best. And then we send them to the automated synthesis lab and start the whole thing again. And ideally, after a few iterations, we get good starting points and then can move on to do what I told you before. All right. So we can have the discussion more about like target discovery, biomarker, etc. And I stop here. Thank you so much.
Very much. Are there any questions? And I should also say that if people online want to engage in the Q&A discussion, you can just use the chat in the call, and we'll read out your questions and pass them on to the speaker. So feel free to engage as well. Are there any questions in the room?
Thank you for your talk. So you talked about earlier about drug discovery. What targets are you currently working on? What terms of this?
So if you're a biotech, you don't want to overly risk yourself. So if you already like have a vision on like how you automate, etc., you don't want to work in a niche or something. So I think we have a big portfolio. I'm actually not the best person to tell you the details, and probably you find more on our website, but we definitely work on cancer targets and on various diseases like on Alzheimer's. We also, I think we have worked and maybe still work on rare disease target, etc. But that's very hard to do at scale. So generally, you have to focus on where you think you have good chances.
Did you also deploy this piece of predictive models, but in drug equipment? So drugs that are already developed, but we might not know their activity in other diseases and other targets. And is there any scope to utilize these?
So yes, so that's something that people do, that companies do. So it's called repurposing, I suppose. There's actually some companies that have completely focused on that. So that's, I think, for instance, Benevolent AI has focused a lot on repurposing. So then I think what people try to do is like usually build like knowledge graphs. So find new links between targets and drugs, or find new links between targets and diseases. So usually I think what is the core piece there is understanding the literature and reading it in a new way. So you build a knowledge graph, or probably now you use a large language model, etc., and you try to fine tune it that it finds connections that are not obvious from the literature. In principle, you could also use these predictive models for such things. So that was your question. The problem is just this space of small molecules is incredibly big, even if they're just drug-like. So it's 10 to the 60 is some number that people throw around usually. So you can't expect ever any predictive model to predict accurately on all of these compounds. It's essentially always on a very small subspace of that. And so if you now look for compounds that have been tested on something very differently, you would need some data that is brings this molecule somehow in domain or gives you confidence that this prediction will be actually useful. But you can also just start generating certain starting points and hopefully you find data from there.
Yeah. You said that you're learning molecular representations from QN and can be looked at. In what form? Like, do you learn an embedding, or is it like a graph, or is it some embedding of the features of the graph?
Yeah, so there's general possibilities. I mean, the kind of models that work very well, molecules are either graph-based or they're language-based, but I believe graph-based models are probably better. Then, oh, by the way, one of the people that really works on this is Ward, who sits over there. And then we essentially, so there's big QM data sets. You can either just use a 2D representation of a molecule, so the topological graph of a molecule, let's say, and learn on quantum mechanical properties, or you learn on certain conformation of the molecule and build it when you want to build a 3D model. So it depends a bit on your application afterwards.
And you use that for both like the generation of molecules and then also the screening?
So this we do at the moment mostly for predictive models. And that the one exception is maybe for things like docking. So bringing a compound into a molecule, because that is very much related to binding.
You mentioned that you use a relatively small amount of data that you kind of generate internally, but I'd imagine depending on the marker you're looking at, there could be a wealth of information already out there that other groups have done. So do you then repeat some of those experiments internally to like sort of standardize it, or do you end up using the literature then in those cases?
So we use literature. Usually, I think we only repeat experiments on when it's like due diligence. Let's say it's competitor compounds that are going to the clinic or something, right? But generally, if it's a lot of data, you don't want to reproduce it because it would be a lot of money. So you just use it, but then usually it's very noisy. It's very low quality data, and you have to essentially filter it. So there's a lot of work then going into filtering to a level where you are confident that it adds value.
And then if you're sorry, just if you're using someone else's data then to generate these models, then because you developed a model, does that mean your company still gets to sort of own the output, or because the input is from other sources that feed into that model, is it a bit sort of muddy and have we figured that out yet?
Yeah, so as far as that, so usually what you use is compounds from patents, and they're public knowledge. So there's no IP that leaks into your model or something like this.
So regarding the generative models, how do you deal with bias in the training set, and do you think come to mind like compounds that have, you know, if you find literature, you can use a lot of actions for certain parts don't have a lot of inaccuracy, how do you deal with that? Then secondly, how do you deal with you know, most molecules that have been published following with some common set of rules, so there's not a lot of variability in the chemical space. How do you deal with that? Do you propagate the same like mistake in your model, or do you account for that and go explore away?
So that's a really good question, because these are actually things that everybody struggles with. Yeah, exactly. So yeah, no, it's a really good question. So the first thing is like, yeah, you can try to compensate for this bias that's all active by trying to find some way to make sure that either you generate net data, so this will happen over time probably by itself, but ideally you find a way to already ground the model in a way that is that it also produces a lot of inactives. So you can try to engineer that. Then there's another problem, is a lot of patent data is also not really clear. Like a lot of times to say, oh, these compounds are in this range or above this threshold, etc. So you have to start building ones that can accommodate for that as well. But generally, I would say, like, you only get that much out of public data, and at some point, your internal data is much more important, and you have to find a mechanism to balance it. So again, sometimes transfer learning, fine-tuning can also play a role in this process.
Given that so much of the expense of the drug design campaign is in the clinical trials, even if we could get an AI to like perfectly design the drug for is there still a route to being able to design drugs for rare diseases?
So, I mean, I'm no expert on this, but what I understand is that for rare diseases, maybe clinical trials are a bit cheaper and easier because probably you have a lot of volunteers, and also it is of benefit that you don't have competitors in most cases, right? So it's a bit of a different situation. Then what you can, even if you, what I present you is about drug design, what you can still do is raise your chances that you make it through the clinical trial, right? And if you do that, that makes everything much cheaper because you don't have to do it only once or twice or 3 times instead of 10 times.
So like high level question, like what's the biggest bottleneck at the moment with regards to scaling like your flywheel thing? Is it like data? Is it architecture? Is it like the automated testing you're trying to do at the end?
Yeah, so that's very clear to answer. It's really the experimental side. So that's like building such an automated platform, right? It's not cheap. So, we wouldn't do that if that would not be by far the biggest bottleneck.
Okay.
All the boss of Eli Lilly was in the newspaper today complaining about the slow speed of the British Medicines Agency, which is comparatively fast compared with the European Medicines Agency and the FDA. Which stage do you think the medicines agencies will be ready to evaluate these alternative processes? Especially in the context that they're very difficult to audit.
Yeah, so, okay. So I can't make much statements about these agencies. It's also really not my background. So when it comes to auditing, like in the end, you have a scientific process. In the end, nobody asks you, how did you learn this fact, etc. In the end, it's like, okay, can you show that this compound is safe, that this compound actually works, etc. So I think that is not very much altered. So what you prove in the end is the compound, right? Not the pipeline that you built to get to that point. And there's still like humans in the loop in this. Will be people discovering that compound. I don't know how much that helps or if it answers the question.
I think you're being a little highly optimistic.
Okay, well, let's see.
All right, in this case, I'm going to suggest, thank you very much, Becca, and thank you for all. All right, yeah, so we're going to move on to the discussion board. So if you had any more questions to the speakers and you didn't get a chance to ask them, feel free to raise your hands again. We'll try to include your questions in the discussion. But just to get us started, so Paula, during your talk, you talked a lot about how it's difficult to diagnose patients with rare diseases and how that's a big challenge when working with rare diseases. Can you speak to what are maybe some other big challenges when dealing with rare diseases specifically? What would you like say are the main challenges of dealing with rare diseases?
So there's for me 2 big points. One of them is that we are used to thinking that we know how the things work, and then suddenly you have a patient which looks like this, but is completely different. So the way to approach it is different. So it's a bit of also having a bit of open mind. And I understand that at the healthcare system, you have a lot of protocols. And this is the way to do things, but not everyone fits in that box. So that's a bit of the issue. And today we're getting a lot of this genetic data, but that means we're also finding a lot of things that we don't know what they are or what they do. And then maybe some of these predictive models that Hagen has mentioned, it could be that say, okay, can you predict me of these 10 variants of unknown significance, which one is more plausible? And then from there, maybe get started. And what I want is streamline the lab part because it's needed. We have to validate, but it would be nice to just validate, not this trial and error continuously.
Yeah, right. And then from the AI side, I wanted to ask, and feel free to, I'd love it to be the question to both of you, in terms of like the equality of the drug targets. Because on one hand, it feels like, you know, when you're dealing with AI models, they're usually not built for a specific target, right? So depending on like which data you train them on, like the technology stays the same in terms of like, it's very much target agnostic or disease agnostic. So it feels like in that sense, you know, you should be able to translate what you do for like common drugs to rare disease drugs very easily. But then I guess a big challenge in terms of like rare disease drugs is that there isn't that much data, right? And most of the AI models are very much data driven. So could you speak a little bit of like how that could be a challenge and maybe how it could be mitigated?
Would you like me to answer?
Yeah,
Okay.
of course.
I can maybe start. So I think the first step that is already a challenge is, let's say you don't have any data on a target. Let's say you somehow understood that this target is the right target, which I guess is not always the case if I understood Paula's presentation. But let's say you got to that point. Then the first question is, can you actually find a drug against this target? So it could easily be that you won't find any molecules that actually bind to this target and change the function of this target. So exploring that is already like very challenging. I believe what can help is that we had like advances around like protein structure prediction. So like if you use something like AlphaFold and all these structures are public, then at least you have an idea how the protein looks like. And then you can maybe either with computation methods or with some AI-related methods explore like how much can actually bind against this target. Where are essentially particular areas where a lot of compounds would bind, and then explore if this binding is actually possible. But
Right.
you essentially have to first do this evaluation, and essentially when you have done this, then you can probably also create starting points from there.
Right. Yeah. So evaluation is the key step. That's what you're saying. Paula?
So from my point of view, it's also to gain knowledge from other fields. So most of the public databases are not built for rare diseases. They're built for cancer or Alzheimer's and so on. But the proteins are the same. So HSP60 is involved in cancer, autoimmune disorder, etc. So most of the research has been done there. And of course, keeping that in mind, but that's how I also do my prediction. So, okay, so people have seen in cancer that this is how it's going on, this work, okay, this looks like my thing, and then I extrapolate. So if I can do that, I guess I can do that too.
Yeah.
So maybe there's not so much in this patient, but there's a lot in other situations, in other organisms, so we can build data on that part. And then just adjust that variable.
Right.
Okay, what happens if there's a particular mutation here, or this level is increased, which that we can do easily in the lab. Quantifying the amount of a protein or the levels, or whether it's giving more function or not, that's the fastest part. So that we can always validate and say, okay, this protein works this way. What happens if the activity is 10 times higher or lower? Then it will help us with prediction afterwards, or at least avoid as part of that.
Right. Yeah. And sorry, I'm just going to quickly check if there are any questions from the audience. If anyone wants to jump in, please feel free. But in case there aren't, so I was wondering, because Paula had the wish list of the things that you wish AI could do. I don't know if like, and most of them were around the diagnosis part. So I wonder, because even for the common drugs, as far as I know, you know, like people spend a lot of time studying the pathways of the disease propagation, and then based on that they choose the targets that we can work with, and we can build all sorts of like knowledge graphs of like targets and diseases and stuff. Do you think, both of you, do you think that that kind of like those kind of methods and methodology, could that be extended to also apply to rare diseases and maybe help with the diagnosis?
I mean, that's what I hope it could be done, and not only with the diagnosis, but also with the prognosis, because it's also not the same to tell to a person your disease means you're going to get sick in this way, and you will also not treat them in the same way, depending how fast it's going to evolve. And that right now is very hard to do. So that part would be also nice. And maybe you could see, okay, this protein is affected, maybe we can try not only this drug, but these combinations of drugs. Because single drugs is still not so difficult for us to do, But if I have to do this and combine it, I will retire before I'm done. So that will be a way to say, okay, and it's probably the solution. It's not a one drug, it's this drug, or maybe improve this protein by doing exercise or things like that. And that's probably where the answer for both common and rare diseases are.
I think, so, I believe like for AI to work, you always need some kind of data to start with. Right? So if you extract it from, like, let's say literature, I think that's one way, and I think we discussed this a bit. The other way I believe could be helpful is when it comes to genomics. So if you have like next generation sequencing data, etc., as Paula mentioned, like if you have enough of this data to classify, okay, what kind of mutations are related to which rare disease, I could imagine that you can build some model on that and give indications, but you need essentially enough data, and I believe it will be very challenging to build a model with the data that's available, would be my expectation.
Right. Yeah.
If I can just follow with the data, because when you were presenting, one of the questions I had is like, okay, there's a lot of genomic data out there, but it's personal data. So like I couldn't, even if I wanted, I could not share my patient results with anybody outside because it's an ethical concern, especially in rare diseases where it's very identifiable.
Yeah.
So how can you access that? Because the data exists. Is there a way that we can do that data follow all the new ethical data protection regulations, or is that a forbidden lost cause?
That's also a good question. I think you essentially need approval from patients as far as I know. And I think that's why also some companies like 23andMe are interesting, because they have the data with the
but that's the US. Europe is a whole new
but exactly, that would be probably the bottom.
Yeah, and I guess my other question is, Paula, you started off with talking about rare diseases, and you gave us a little bit of an intro on like what they're about and what are some of the challenges that you face when you work with rare disease patients. And then Hagen spoke about AI methods and the whole like array of different applications and different methods that we use when dealing with like this drug design pipeline. So after this little bit of like a knowledge exchange session, do the two of you have any thoughts on how the two worlds could be combined and how like AI could be used for rare disease, finding drugs for rare diseases and or helping with diagnosis of the rare disease patient?
So I believe the area where probably AI methods are already used and where it could be very useful is around really scanning the public knowledge and finding indications, comparing diagnosis, etc. And I think that's probably where I feel like your talk was aiming at this, and I know that companies are doing this, and I think Accenture is doing this as well. So.
Right. Yeah.
That even if it seems like a small job, it's very time consuming, and there's never a way where you can screen everything.
Yeah.
Because sometimes you have a paper from the 80s and that's where your answer is, and that you never get there. It's impossible. And also the machine, I guess they will search in a different way that I will do. So maybe we'll find other things.
Mm-hmm.
I also think that there's a lot of data. I cannot process all the data I have. So from this, from both of the studies I have, both genomic, transcriptomic, proteomic, and metabolomic data. And I combine them in my way, which is probably much simpler than what AI can do. So I think they can also take that, okay, maybe it's just one patient or a small group of patients. There's a lot of data, and a lot of these patients are follow up very much. So they maybe have a longitudinal data. So I think the data is not about how many people, but how much from few, with the limitation that means. But I think there's something there to work, and prediction is what I think for me is the most interesting.
Right.
To give a more clear answer, also to see, okay, from these patients, these are the ones I need to focus today. This maybe in 5 years.
Right. Yeah. So this kind of like scanning of the current like knowledge that we have in the public domain and extracting the information and building links between like what we have and kind of like making it easier to use the data that's out there. Right. So like information retrieval.
And data analysis too,
Right,
because that's a bit of the issue. So I know in some of the hospitals in Denmark, they're trying to build a pipeline because the genomic data is humongous.
Yeah.
So unless you find your answers in the first classical ways of doing it, then you have a lot of data that you don't know how to process. So
Yeah.
maybe one of these models can help to do this kind of ranking. Say, okay, you have to validate, but maybe start with this few variants instead of not having a place to start.
Yeah.
So that for me is also very interesting.
Yeah, I think following up on this, I think like combining different modalities is something that can be very powerful. So combining genomic data, transcriptomics data, and maybe, I don't know, extra visual data of I don't know, bone structure or whatever. But I think the problem is you also have to then collect the data in a consistent way. And I think while there's a lot of data, combining it in a consistent way is often very challenging.
I would say that would be a bottleneck. That's
So you have to generate it essentially with that in mind.
Yeah, right. Yeah, that makes sense. Oh, what
I
question?
guess, yeah, just to also ask about it. Are there sort of easy things that sort of the current experiment can do to extend the usefulness of data they generate for disease?
I mean, there's some experiments, especially the functional analysis that are very easy to do because you're using models. If you do something in bacteria, that's easily 24, 40 and so. So that and mass spectrometry that can be scaled up very much. The issue is usually when you go to more functional things, when you are using human cells and you just start putting a lot of things. So of course I measure two, but they're two things happening at the same time. So that's a bit maybe where I see a more limitation. How much a protein works or not, that's very easy to do because that we have been doing for the last 30 years. So there's a lot of assays and knowledge in that. We're just getting a bit lost because people is retiring. So we don't do those old school things anymore. But that is, I think we can add a lot of things. And then, I don't know, for me, the prediction of saying, okay, we know how this works. What happens if you increase this 10 times? It's very hard for me to predict, but then I guess some of those models can help us predict what happens if the protein works faster or slower in the other 2000 process.
Isaac?
I don't know if I know, but are there any antibody drugs for diseases? It would be challenging, I guess, for like all it.
There are some, especially for autoimmune disorders, and there are some of the ones with mitochondrial dysfunction that they are targeted in that way, because mitochondria can generate autoantibodies. So in some of them, that's a symptom, but those are very expensive therapies. So it's very hard to prove the usefulness because it's those plasma replacement and all of those. So it takes, and it's also not very nice for the person. So we do it if it's needed, and I know some cases, but I also know that the physicians are not very fond of this.
Yeah, question.
How do you think we incentivize researchers to spend their time with this? Do you think that making it easier is going to be enough?
Money.
My thoughts exactly. That's
I think it's, I mean, there's a lot of problems where, let's say, yeah, I think as a company, it only works if you can financially survive. You can't be a charity. So if there's an incentive, let's say from a public body, etc., I think that makes a big difference.
So this is like kind of thinking in the same sort of like train of thought, because I've heard this argument being used around like how you know, our technology and the processes that we use to find new drugs in general, that's improving constantly, but the rate at which we're actually putting drugs out in the market for patients to use, that's constant, if not declining. So even though there's all this, you know, improvement and progress, there isn't that much output out of it. And one of the hypotheses that people say is the reason for that is that maybe we found most of the low-hanging fruit, and it's hard to find new, like the new drugs that we're maybe about to find are just hard to find. So could it be that one of the motivators for looking into rare diseases is that maybe we haven't found all the low-hanging fruit there yet because we haven't explored that space as much. So do you think that could be like one of the motivators also to like look into rare diseases a bit more thoroughly?
For me, it makes sense because especially when you talk about rare diseases, you're talking about something a bit extreme, which usually is not common to find, but it makes some things easier to detect because you're in this more black and white situation, not that much the grays. So it could give us some knowledge also. So some of the things we do are applied also in cancer. So you can also have the things like, okay, yes, we're helping these people, but we're also using them as a model for more common disorders like my diabetic patients. They're not the same as the type 1, type 2, but there are some things in common. So what we gain here can also be applied here. So that I think for both private and public, it's also an incentive. And then again, it's just about cost effectiveness, also for the public sector, because even though we're not about the money, we all have to pay rent and everything. So it's a limitation still. So that's and at the end, they prioritize what it gives more for the money. That's
yeah, I think usually these arguments come after looking at the explosion of costs over time in the pharmaceutical sector. So it's really like a post hoc explanation and putting your bets on it, like saying, okay, maybe something is easier, so let's try it. It's very difficult because I think in the end, it's all a question of how much risk is there compared to reward. And I think the smaller you are as a company, the less risk you can take, etc. So I think that's essentially like you have to estimate this, and hopefully you're right.
So would you say that there's probably more risk associated with working with rare disease targets?
I think so. So I think why a lot of pharma companies, a lot of drug discovery projects are on targets where other people are already working on, right? And that's essentially just, okay, everybody's already working on it, so it's maybe less risky to work on it. And that has also to do with knowing that the target is actually related to the disease, so I think you can really see that a lot of it is about de-risking, and I think when people go for first-class targets, meaning a target that nobody has worked on, there's always an intrinsic risk, but at the same time there's less competition, so you have also advantages.
Yeah, that's true.
And that's also where the public and the private can meet. So some of the things we do is take those risks. So with this content, they were very sure of the things, but they were not interested in the ones. So we have no idea how it will work. So, well, but we can test that. And now that we know, then they can take it into the pipeline. And for us, it doesn't matter whether they take it into the pipeline or not. So that happens a lot, especially in Denmark. There's very close private and public collaboration. And most of it is about de-risking. So we think this is working this way, and then we can do that part. And then the company has a bit of the protection.
Yeah, makes sense. I think I saw a question at the back.
This is perhaps less about drug discovery than treatment discovery. Could you comment on drug therapies, particularly where you might need a better understanding of how a drug acts in a whole organism, and that could be like more tricky for rare disease where you have kind of less information about.
I mean, I think that's difficult in general because it's also there's a lot of person-to-person differences. So especially if you look at the elderly, they are taking a billion things that we have no idea how they work together. So there's a lot of research going on into that. And I think it's still, even if it's a common disease, it will be problematic because we don't metabolize things the same way. So I think it's a general problem. And probably the cure for a lot of diseases or the best treatment is a combination. And what about unmeasurable things like exercise or social activities? There's a lot of research showing that those are beneficial too. How do we combine that? And I don't know if AI can put that together, but that's also the next situation. It's not just about the field, it's about all the other things too.
Yeah, absolutely. So I wonder, 'cause, you've spoken about personalized medicine. Exentia also, I guess it's a bit further from what you do, Hagen, but Exentia also does like personalized medicine. I wonder if there is some sort of overlap that would be interesting to explore. Like so you can speak maybe a little bit more. You definitely know more than me about this, but how the personalized medicine, the precision medicine at Exentia works, and if you see like how it could be applied maybe to rare disease patients or rare disease diagnosis.
Yeah, so I didn't bring it up because it's a different domain. So it's for cancer treatments. So what we do essentially, we take
opposite of rare disease.
Yeah, exactly. So we take like biopsies of cancers from patients and do experiments in the lab directly on those cells, and then use this as a signal to say, okay, A as a signal for this drug works well or not, and B as a signal for this patient should be treated with drug A, this patient should be treated with drug B. So precision medicine. But I don't know how I would do the same thing for rare diseases.
I mean, the experimental part is kind of the same thing. And when we talk about cancer, we of course think about the common cancers, but there are also rare cancers, which are actually more deadly than the common. So that happens a lot. Most of the bone cancers are rare cancers. So most of the children cancers are usually rare. And at the end, taking a cell and putting it there, that's enough. What I don't know is whether doing this precision medicine for this one person, how it gets covered, because then Exentia doesn't do the treatment. I guess they feed the information to whatever hospital and they take the treatment. It's just how it works in Denmark. And that's the difficult part, because it's also a difference regarding patient lobbying. So the cancer lobby is very strong. So then they put a lot of pressure into it and very quickly it goes to the media. So there's also a lot of different levels of involvement in that sense. I think that's more the issue from my point of view, from my little knowledge of AI, than the part, because the cell is a cell at the end.
Yeah. I wonder if like a similar technique could be used for diagnosis. So like in this case, like it's applied to identify like a successful treatment for a patient. But I wonder if you could like in a standardized way, like test like really quickly also on like a small biopsy, like a number of responses, and based on that, you could get the diagnosis faster, maybe. I don't know.
Yeah, so what you can certainly do is look into phenotypic differences between patients. So what we do at the moment is look at biomarkers. So can we stratify patient populations by the difference in how the cells look like, or how the cancer cells look like, etc. But in principle, you can also ask, do immune cells look different, etc. And I think that's an interesting direction to and this you could also use for diagnosis.
Yeah, so it would be like a vision task, right? Like kind of like scanning for differences. Yeah, I think that could be interesting.
And you can also correlate again like genetic information with visual information of cell images, etc.
Yeah, and maybe then, because it's very easy for us to classify the extremes, the most asymptomatic and the most severe, but all the ones in between is very hard.
Yeah.
So that could be a way to classify them and also kind of prioritize the ones you suspect that they're going to be sicker or worse, and you could put more effort into that.
Yeah.
And then just also, sometimes we forget that it's worldwide. So maybe there's not a lot of patients in the UK, but it could be that if you take it all together. And in that sense, it could be what Hagen was mentioning, that maybe nobody's working in this rare disease. So maybe there's only 5 patients in the UK, but there's 5 in Denmark, and then it builds up.
Yeah.
That could be a way to also get some traction.
Yeah. Any other audience questions that I've missed? Any questions online? No? Okay, great. I think we'll probably have time for like one more big question. I don't know. Do you have any concluding thoughts? Because I was thinking now that I wonder, because you had this diagram of, like sometimes even if there's the same gene mutation in patients, you can go from anywhere from asymptomatic to really extreme symptoms. And I wonder if it would be possible to like build a model where you input a bunch of like random features that would be like some sort of like symptoms of a patient that you would maybe not necessarily like think of when you're like studying a patient as a human, and you would look for correlations between like, I don't know, like, you know, those features and then the phenotype of the disease, and maybe that way you could like gain a bit more information about the disease as well. Yeah, but I don't I don't really know how that would work, but I'm just thinking in terms of like how little we understand from like, you know, how the disease is actually going to manifest itself. Yeah, but we have like 2 minutes left, so I wanted to give you a little bit of space to maybe share any concluding thoughts if you have some.
For me, it has been very interesting because it's also to see another perspective. And I definitely see an interconnection of the things so where we can benefit both sides. And then, of course, there could be a lot of other issues out there, but at the end, it's just how to make the best of what we can. And that I definitely can see AI as a very interesting tool for us to do much more than what we can do today with less.
I think it's a really interesting domain, and I wish there would be more opportunity to really think about it and work on it. I think there's a lot of potential.
Yeah, absolutely. I think so too. All right. Well, we're basically out of time. So thank you all for coming and joining and engaging. And thank you very, very much for both the talks and the discussion. And thank you for coming all the way.
That's
very much
wonderful.
appreciated. Thank you. And I think we can thank our